The last presentation I showed you a lot of background information on the science that led to the discovery of the glycocalyx, the imaging methods in academic centers and in research settings, and the implications of damage of the glycocalyx for vascular function. Just to summarize the most important findings of that first presentation, we showed you that the vascular system is not only composed of the large vessels, the arteries, and the veins. 99% of your vascular system is actually the smallest vessels, the microscopic capillaries that you only see when you use big microscopes and see the individual red blood cells flowing through these capillaries. And if you zoom in with a microscope to see one of these individual capillaries, I demonstrated that the red cells flowing through these individual capillaries, they never touch the wall. There's always space between the edge of the red cell column and the luminal surface of the endothelial lining that makes up the wall in these small capillaries. And then if you zoom in on that space between the red cells and the luminal surface of the vascular wall, that this space is actually filled with a polysaccharide matrix that we call the glycocalyx. So here again, you see a cross-section of a small capillary. And on the right-hand side, a larger magnification. Here on top would be where the red cells are flowing. Here's the endothelial cell lining of the vessel wall. And then produced by the endothelium, you have these bush-like structures, these bush-like polysaccharides of heparan sulfate and hyaluronic acid, produced by the endothelium and bound to the membrane of the endothelium, and then sticking into the lumen of the blood vessels. And in this polysaccharide matrix, or glycocalyx, binds a lot of plasma proteins and fluid that binds to the proteins, forming a very stable gel-like protective inner coating of your vascular system. Then we also showed in the previous lecture that this glycocalyx protective coating is very closely related to endothelial function. So any function of the endothelium and vessel wall function that we know is mediated by that glycocalyx coating on the membrane of the endothelium. So I showed you that blood flow bends forward these polysaccharide structures, and those structures amplify the so-called shear forces of the flowing blood, telling the endothelium to start producing nitric oxide. Also, these glycocalyx polysaccharides bind important enzymes like superoxide dismutase that capture and activate the oxygen radicals that are locally produced, thereby preventing the damage that those oxygen radicals might be able to do in the absence of this enzyme. At the same time, the tight nature of the polysaccharides within glycocalyx prevent leakage of fluids and proteins and larger molecules like cholesterol. And also the shear dimension of this glycocalyx up to a micron or longer also physically prevents other circulating cells like platelets and leukocytes to get in physical contact with the small adhesion molecules that are only nanometer scale long that are produced and presented on the surface of the endothelium. So a healthy glycocalyx prevents the sticking of platelets and prevents the binding of the leukocytes, preventing local inflammatory response.